Hey everyone, welcome back to Affordable Tutor, where we make education simple and exciting for everyone. In today's video, we are going to learn how you can find RS or single nucleotide polymorphism for your gene of interest. So for the purpose, we will be using GnomAD database, which is called Genome Aggregation Database, that brings together exomes and whole genome sequencing data from several studies reported in different databases and in different projects on internet. So here we can search like GnomAD, and here we have the first one URL. We'll be using the default version that has been selected so far, and you can see we have different assemblies and different versions that depend on your type of research you're conducting. But here you can see in the search bar you can search through gene, regions, or variants. We'll be using gene EGFR, and we'll be click on it, and here we have the information regarding our gene, but we are here for the RSIDs, so we'll be going down for it, and here we have it. Like, these are the RSIDs, variants, and other informations, variant effect, predictors, annotations, and so forth. Here you have a filtering abilities that you can filter so far. Here you can search it for yourself, and here we have like you can configure your tables accordingly, like RSIDs. If you want these to be up or to be down, you can just drag in up and down, or you can use these arrows also. Now it's safe, and here it will be. But these are not the total RSIDs for your genes. Like here you can see in the node, only variants located in or within 75 base pair of coding exomes are shown here. So to see all of your RSIDs, so you can just click on the region view, and it will be loading for some time, so be patient for it. And in the meantime, if you didn't subscribe to our channel, so please do it. And here we have the loading. The RSIDs has been loaded, and yep, these are all the SNPs, all the RSIDs that has been reported so far for EGFR in GnomAD. So you can just click on export variants to CSV, and it will just give you download options here, and you can just go and use it for your research purpose. So that was all about today's video, and if you like this video, please give it a thumb up and give us a subscribe. And if you have any questions regarding anything, just let us know in the comment sections. We will try our best to help you out with that. So see you in the next video then.